This is a University of Otago podcast.
All right, so we're going to have the next talk in the session, and that's from Stella Cameron and Louise Parr Brownlie. They are going to be giving us insights into Parkinson's disease.
All right, so good afternoon, everyone. I'm Stella Cameron, and I'm a PhD student in the anatomy department, and this is my supervisor, Dr. Louise Parr Brownlie. And we're going to be talking today about Parkinson's disease, as this is what our research is about. So I'll be talking about what Parkinson's disease is and the current limitations and treatments. And Louise is going to be talking about some upcoming treatments. So if you've heard about Parkinson's disease, it's likely maybe associated with Michael J. Fox or the late Muhammad Ali, who have greatly increased the awareness of this disease. So in New Zealand, about 1% of the population over the age of 60 are affected with Parkinson's disease. And the number of people with Parkinson's disease is expected to greatly increase in the coming years due to an ageing population. And so it's really important that we are continuing our research in Parkinson's disease and developing more effective treatments. So people with Parkinson's disease have very characteristic motor symptoms. So here we can see a deficit called bradykinesia, which is the slowing of movement. So you can see in this patient's left hand that this movement is greatly slowed or reduced. And akinesia is the difficulty in movement initiation, and you can see this in this very shuffling, limited gait of the patient. And tremor is the involuntary shaking of the limbs, and that can also affect the head. And there's other motor symptoms such as postural instability as well as non-motor symptoms. So why do these occur? Parkinson's disease is a progressive neurodegenerative disease, which means that we're seeing a progressive loss or death of neurons within the brain, and specifically the neuron that's lost is the dopamine neurons, and these produce a signaling molecule called dopamine, which is really, really important for movement as well as other behaviours. And a large population of these neurons are located within the brain, roughly around here, called the substantia nigra, which you can see in this midbrain, human midbrain here, and you can see it's greatly reduced in the Parkinson's midbrain. And with this loss of dopamine neurons, you have a reduction in the amount of dopamine in the brain. And this is really important, as this is what is leading to these movement deficits. So what can we do to help Parkinson's disease patients? Currently, there's a drug available called levodopa, which is a dopamine precursor, which can be taken up in the brain and used to synthesize dopamine. And when it was discovered, it was like this magic drug, like, yay, we've increased the amount of dopamine, we've solved the problem. However, within a few years, Parkinson's disease patients start developing quite severe side effects to the drug. You can see in this video here what's called dyskinesias. And you can imagine that these movements, these involuntary movements, are quite difficult to live with. And this has led to the development of deep brain stimulation, which is the implantation of electrodes deep down into the brain in an area called the subthalamic nucleus to stimulate this area to alleviate these motor symptoms. And so here we can see it in action. So this is a patient before the deep brain stimulation was turned on, and this is a patient afterwards. So you can see this really dramatic effect of this treatment where those movements are greatly reduced and he can quite effectively perform this motor task. So deep brain stimulation has really greatly improved the lives of many Parkinson's disease patients so far, but it's not without its limitations. So for one, it's a very invasive treatment, and that therefore comes with its own risks, and it can also, patients with deep brain stimulation can result in some side effects. So, for instance, you can have some behavioural problems where where a patient may become more, their mood may change a little bit. So I'll now hand you over to Louise, who's going to explain a little bit more about why these effects occur.
Kia ora koutou. So to orientate to what we've got in front of us here, we're looking right at the tip of the deep brain stimulating electrode, and we have 2 points of contact, which are the black little squares here, and we're going to pass electrical current between those two contacts. And what happens is that current spreads out, and if it keeps on spreading and spreading and spreading, it will actually activate cells in a neighbouring brain structure, and that brain structure may have quite a different function. And so as that current spreads, you start to bring on these side effects. So we've been using a new set of stimuli where we use light to activate brain cells, and brain cells don't usually get activated by light, so we have to do a whole heap of manipulation. So what we see in this diagram here is the blue light coming down. This cell, we've modified it. I'm about to explain how we do that in just a second, but you can see these blue circles. And only the cells with the blue circles will become activated and not other ones. So the purpose or the advantage of this technology is that it is incredibly specific, both to the type of cell that we activate, because there are many, many different types, and also to the structure, the nucleus within the brain that we do activate too. And of course, we're hoping to reduce side effects by doing that. The way that we get these light activated or make cells light activated is that we're going to use a virus. And viruses on the outside of them have a characteristic pattern, and they will have a target cell within the body that they go to. And when they get there, there is a response that always occurs. So the virus gets to the target cell, the virus's genetic information goes into the target cell, and we experience this all the time when we get exposed to a virus and we end up with the flu symptoms. So it hijacks the genes and makes us create different proteins, and therefore we end up being ill. So what we do is we're going to use the virus, the outside of it, as a packaging and a delivery system. We take out the virus's genes and we put in the genes that are going to control the light stimulation. That genetic information, what we actually have here is the cell membrane, is these brown circles. This blue structure is a protein, a channel or a gate, and whenever we have blue light coming into the brain, a little pulse of light will open this gate, and the ions can now cross the cell membrane, and the cell can become active. So we can just pulse light in and activate the brain as we want. The genetic information also contains this red flag. This is really important because at the end of our experiments, we want to know which cells the genes have got to, or we're having these light-activated genes expressed. And on the right-hand side, we have some brain tissue where a cell has been altered, and you can see that is indeed red. So what we've been doing is using this technology to try and develop a new way of stimulating the brain to get improved functional outcomes. So we use a rat model of Parkinson's disease, and I'm going to show you two videos. The first one on the left, the animal is Parkinsonian, and just like Stella showed you right at the beginning, he's not going to be reaching at all. But he can move around in the cage at will, and what he will want to do but is unable to do is to reach for a Cocoa Pop. He likes those. And he will do those at will if he was able to move around. So this rat, you can see his head bobbing up and down. He can move, but he doesn't execute any reaches during this period of time. The second video is the same rat about almost 20 minutes later, and we've been flashing little blue pulses into the brain, and you can see that one pulse is sitting there. You can actually see it just sitting above the head. And now as the rat, after 20 minutes of stimulation, it's starting to reach through, and you can see the paw flash through in this region just here, and then he'll pause and he'll eat. So by flashing light into the brain, we could recover movement. What we're aiming to do is that this is going to be the next generation of deep brain stimulation, but it's going to be much more specific and have less side effects. And of course, a rat brain is very tiny. So one of the things we are doing at the moment is changing the technology so that it works in the much larger human brain. A second technology that is new is, an old technology is to use lesion. So the patient comes in, it's a neurosurgical technique where a frame is fitted to their head, they have an MRI scan of the brain, and they target to that subthalamic nucleus, that structure in the brain that Stella talked about earlier. And they'll put this probe in, and they'll just burn a little hole, and it does improve symptoms. It's very effective, but it's also very invasive. The latest technology that is available is something called MRI-focused ultrasound. So here is a person, they have one of those frames around their head, and it almost looks like a helmet. That helmet is actually part of an MRI scanner, and also within that, it generates ultrasound beams. So on the right-hand side, here is our target. This device generates 1,024 ultrasound beams. Each one of those only has a tiny bit of energy, but where they meet up, the energy is intense, it heats up the brain and it burns a little hole in there. The wonderful thing about this is that there's no surgical procedure. The person walks in, it's a day surgery procedure, about 2 hours later, they're already having improved symptoms and they walk out again. And if necessary, because it's non-invasive, they can return in a couple of weeks, couple of months later, if the effect isn't as good as what the surgeon expected. So what we've talked about today, we've talked about some of the limitations of the current treatments of Parkinson's disease. We've highlighted 2 future ones, one that we're developing up in the lab and one that is available in only 5 sites around the world. And to finish up, we would like to thank the members, the current and past members of our lab. There's been a lot of research to be able to do this light stimulation technology. We have a lot of collaborators here in Otago and up in Auckland as well, and our funding sources.
Cool, it was very interesting. Thanks for that. Does anyone have any questions about the brain? I have some. I constantly have questions. So for those first couple of videos you showed, more of a question, well, 2 questions really, but about the disease progression, they seem to have a unilateral progression. So initially it presents on one side. Is it usually the left? Was that just because it was 2 videos? I mean, it's a small sample size, but...
Yeah, it can vary depending on the patient, but often it will be one side more affected, and then as it progresses, they'll both become more affected.
Okay. Yeah. And so does one side catch up to the other side, or is one side always worse?
I think the original side is usually worse, and usually if you get the dyskinesias, it's worse on that side as well.
Okay. Does anyone else have any questions? So I've got more. That's all. Okay, so the next one was, because my work is on dopamine receptors, and obviously humans have a lot more dopamine receptors than honeybees, but is there an association between, say, some autoreceptors or some dopamine receptors in the brain that, or splice variants or whatever, that associate more with Parkinson's disease than with others? Or is it just, I don't know, this may be outside of the things that you do as well?
No, it is outside our expertise, but no, I don't think so. The thing with most Parkinson's patients is we have absolutely no idea why they end up with Parkinson's disease. Less than 10% have a genetic predisposition. The remaining people, there's just been small mutations in their genes that have caused it, but there are some risk factors, and some of those are things like environmental factors, being exposed to pesticides and also to heavy metals.
All right, okay, cool, thank you very much.